Okay, this lecture we're going to be discussing the central nervous system. So the central nervous system is kind of a branch, a division just of our nervous system in general. We'll see that we'll have a central nervous system and we will have a peripheral nervous system. Now these images that you can see on this introductory slide, you can see a large brain. You can see some other accessory structures. You can see a cerebellum right back here on the posterior aspect of the brain. You can see this medulla oblongata region, a pons, spinal cord coming down. And then on this inferior image, you can see some spinal nerves that are, some cranial nerves here that are coming off of our brain. And so we'll take a look at this very closely. We'll see that the central nervous system is composed of two main structures, and it's these two structures that we just talked about, the spinal cord and the brain itself. Everything else coming off the spinal cord or off the base of the brain will be part of our peripheral nervous system. We'll talk about that separately. So to start off talking about our central nervous system, we need to understand kind of how we get our central nervous system, or rather just our nervous system in general. And so as we are developing, after we have been, after an egg from a female and a sperm from a male have united to form a zygote, we undergo division of cells. We go from one cell, we go to 2 to 4, 8, 16, 32, 64, and so on, until we get this large kind of ball of cells, essentially, and it's somewhat hollow inside. Now, within this ball of cells, and this will be termed a blastocyst, you get the development of different things called germ layers or embryonic germ layers. You have one called an ectoderm, you have a mesoderm, and you have an endoderm. And each of these embryonic germ layers are going to further differentiate, and the cells will divide and become different structures that we see in our adult form. So we'll focus on our ectoderm here, discussing the central nervous system. But understand that the ectoderm can also leads in the development, or leads to the development of our skin as well, our epidermis. So our epidermis and our brain and nervous system are actually related in the sense that they both came from the same embryonic germ layer as we were originally developing. Now there are regions in this blastocyst that we just discussed that become kind of thicken, and this is going to be termed the neural plate. And we'll see this neural plate, and then we get something known as a neural groove. Now this neural groove is an area where our neural plate begins to kind of fold back and invaginate upon itself. And when it's termed the neural groove, it is open. It is not completely closed. It doesn't make a complete circle or tube. Once the two edges of the neural plate actually come together and fuse and touch, we change the name from neural groove and we go to neural tube, because this tube is now a complete circle. It's completely enclosed and is now no longer open like we see with our neural groove. So this is a picture of our blastocyst here. So you can see a couple of different cavities, amniotic cavity and yolk sac, and then these three different colors, this blue, this red, and this yellow here, are our three embryonic germ layers. So you can see ectoderm on top, mesoderm here in the middle, and our endoderm down below. And again, each of these will divide and further differentiate into different structures. Like I said, our ectoderm will develop into things like our brain, central nervous system, and our skin. We have our mesoderm. It will differentiate into things like different types of muscle tissue, so cardiac, skeletal, smooth muscle tissue, as well as our blood. Our blood is derived from mesodermal tissue. And then our endoderm, this last one, the one that you can see in yellow here, this is going to differentiate into different structures like the thyroid cells, pancreatic cells, so very glandular-like tissue, as well as the cells that make up our lung. Okay, so there are lots of other things. The endoderm also contributes to the inner lining of the digestive tract. So the epithelium lining the digestive tract is composed of of endodermal tissue. Our sex cells are derived and originate within our yolk sac from endoderm as well. And so when we're going from the neural groove to the neural tube, this is something that we'll see. So this would be as if you're looking kind of at an anterior. You can either think from an anterior or posterior view of our blastocyst here. So if we see this, it'd be as if we were looking straight on at this ectoderm, and that's kind of what we see. So we see this large neural plate, and we have a couple of different cell populations. So we have ectoderm over here. We have something that will become something we'll call our neural crest, and then we have this middle layer here. And so what you see begin to happen is these two areas of this neural plate begin to fold back and kind of invaginate upon itself. And so at this point, we're calling this a neural groove. And then when these two edges actually touch, they fuse, and we get something known as a neural tube. So it's completely closed. This process is known as neurulation, and it begins near the end of the embryo, or toward the what you can sometimes refer to as the tail or the caudal end, and it progresses toward the head. And so not only can you see this happening in the embryo, but when you see it, you have an idea of the timeline of development, because it starts near the inferior aspect of the embryo and progresses toward the head. I'm sorry, it's actually going to be progressing toward the tail region. I apologize, I misspoke. But what we have here is now we have this overlying layer of ectoderm. We have these cells that we'll call our neural crest cells that will develop into and differentiate into our dorsal root ganglion of our peripheral nervous system, so a lot of our sensory cells. And then we have this neural tube. And this neural tube, you have to picture it in a three-dimensional manner, is going to be running the length of our embryo. And there will be areas where this neural tube will kind of swell out and form these distinct regions that will become different parts of our brain. So the different parts of our brain that we'll see are our forebrain, midbrain, and hindbrain. And these are termed, forebrain is called prosencephalon, midbrain is termed mesencephalon, and our hindbrain is termed our rhombencephalon. And so this is what we see, these primary three brain vesicles. You see this in the embryo, and then as the embryo continues to develop and mature, you see differentiation of some of these vesicles into further components. Okay. Now we retain part of this neural tube, so this neural tube that we formed right here, we actually keep part of it in our adult form as our brain ventricles. And we have ventricles, we have 4 ventricles in our brain, and within these ventricles we have something known as cerebrospinal fluid, or CSF, that circulates throughout the ventricles and then enters the area surrounding our brain in the subarachnoid space, as well as the subarachnoid space around our spinal cord, and serves to kind of protect and cushion these different structures. And so you can see some areas like the forebrain and hindbrain have further differentiation. So telencephalon, diencephalon, metencephalon, and myelencephalon, and we'll talk specifically about those and what adult structures arise from those. So this image here is showing a lateral view and then either an anterior or posterior view here of the primary vesicles. So brain vesicles. So you can see one here. This is going to be our prosencephalon, our mesencephalon, and our rhombencephalon. And then as we continue into about week 5, so about a month after conception, we begin to get even further differentiation. So from our prosencephalon, we get 2 main differentiations. We get a telencephalon and a diencephalon. This will make up the actual brain itself, so the cerebrum, and then the diencephalon includes things like the thalamus, hypothalamus, and epithalamus. And we'll discuss what specific structures and what relevance these locations have as we progress. Now, the mesencephalon does not. There's no further differentiation in it like you see in the prosencephalon and the rhombencephalon. So it just stays, and in our adult structure we call it the midbrain. And then our rhombencephalon divides further into the metencephalon and the myelencephalon. This includes things like the pons and the cerebellum, the metencephalon, so that cerebellum that we saw in the very introductory slide. And then our medulla oblongata is going to be this region just beneath the pons that we'll term the myelencephalon, or develops from the myelencephalon. Okay, so lots of terms here. Anytime you see the word encephalo or cephalon, if you remember, cephalic, the cephalic region is referring to the head. So all of these terms that have this ending encephalon or cephalic or cephalo, we're referring to the head. Okay, and you can define a head as something having a concentration of neurons in one area, as well as a concentration of sensory neurons. And so we have a head, we have a concentration of neurons as our brain. So we have, on the order of billions of neurons in our brain. And then we also have concentrations of sensory organs within our head as well. So we have eyes for our special sense of vision. We have our tongue and taste buds for our special sense of taste. We have our olfactory nerve and our nose for our special sense of smell. And we have our ears also located on our head for our special sense of hearing. And so a head is defined by those things, a concentration of neurons at one region, as well as the concentration of many sensory organs in a single region as well. So the ventricles of the brain, as we discussed, you have 4 total. You have 2 lateral, and a third, and a fourth. Now, circulating within this, we have our cerebral spinal fluid, and it provides some buoyancy, so it allows the brain to be maybe a little bit lighter and not as heavy within the skull. It has a protective function as well. Whenever it circulates around the brain, as well as when it circulates around the spinal cord, it serves to kind of cushion and protect. And then also some chemical stability functions in terms of regulating ions and different materials that you may find within the CSF. And the CSF, cerebral spinal fluid, is formed from cells that we'll see in a little bit called ependymal cells. And a bunch of these ependymal cells kind of aggregated together form something known as the choroid plexus. And this choroid plexus is going to filter blood. From that blood, it removes different ions, different things, and the finished product that comes out of it is our cerebral spinal fluid that will then circulate in our ventricles and around our brain and spinal cord. Okay. Now, cerebral spinal fluid in our central nervous system has some clinical relevance as well, where in instances of possible meningitis, a lumbar puncture, sometimes referred to as a spinal tap, can be done, in which a needle is inserted into the subarachnoid space. And when you insert the needle into the subarachnoid space, some CSF flows out. And by collecting a sample of CSF, you can then take that, analyze it, see if there is any bacteria. You can have bacterial meningitis, you can have viral meningitis, and you can test this sample and see if there are any of those viruses and or bacteria present. And we'll talk later about our meninges, which is meningitis is the inflammation of meninges, and we'll talk specifically about the different layers of the brain, the different meningeal layers, and how they are distributed around the brain as well as around the spinal cord. So here you can see some of those ventricles. You can see a lateral here. You can see 2 lateral here from this anterior view. You can see a third here, third right here, and then a fourth way down here. Now, the way that these connect to each other, your 2 lateral ventricles connect to your third via something known as the interventricular foramen. Interventricular meaning between the ventricles, and a foramen meaning an opening or a hole. So this is the hole between the ventricles, between the lateral and the third. And then the cerebral aqueduct drains CSF from the third ventricle down to the fourth. And then you have these apertures. You have 2 lateral and 1 median aperture that allow CSF to exit the fourth ventricle and enter into the subarachnoid space and circulate around the brain and spinal cord. So here's another image showing that 2 lateral, 1/3, and a fourth, along with the cerebral aqueduct and an interventricular foramen. We'll talk specifically about kind of where you can find these in the brain and how you're able to identify them during a dissection. So we will stop there, and we'll pick up with cerebral hemispheres and some other terminology in part 2.